Morning guys, welcome to another instalment of Biology with Miss Cherry. This is the start of Module 8 and this unit is looking at genetics and population genes and how to use things like genetic sequencing and things like that to have a use. To how to use it as maybe forensics, maybe how to use it for paternity studies, things like that. Okay, so last year in Module 4 you started to discuss genetics, and you can take a look now, so pause the video, have a look in what's happening in those pictures below. So we've got a gene, or we've got a sequence of nucleotides, bases, and see what happens in each of those pictures. So they're separate, and what happens from step 1 to step 2. How can you describe them, and what is the effect of those changes of the sequence in the downstream kind of biological activity? Okay, so pause, and when you're ready, play again. So for today, we're going to take a look at gene mutations, and this is lesson 8.1, so we're going to go over a recap of module 4. We're going to take a look at the types of gene mutations, how they have an effect on the polypeptide sequences, and then to explain why some mutations are harmful and some aren't. Okay, so we can see here that we're linking to module 1, which is our biological molecules, and module 4, the stuff that you did about mutations already. Alright, so let's get in. Now, to recap what you should know already, hopefully, you might not, though you might not be too confident on it, DNA makes up our genes. So we've got specific base sequences that are found on DNA that together code for a protein. And the way that the body reads the DNA is found in triplet sequences called codons. So that might be an A, for example. And these codons work together with the ribosomes to produce an amino acid. Okay, and we're going to take a look at those amino acids in a second. Now, the amino acid goes to make a protein, so the amino acids bind together, they undergo a condensation reaction, okay, thinking back to module 1, and they're producing a peptide bond to bind them together to give us a protein that's relevant to the gene function. So if I'm looking at a gene for eye colour, for example, then the amino acids would be for something like pigments. Yeah, so they're not going to go on and make hair texture if it's a gene for eye colour. Okay, now the DNA is degenerate, and you need to know that keyword because it's generally one of the ones that is underlined in the exam. So degenerate doesn't mean anything bad. It means that more than one codon, so more than one triplet sequence, makes the same amino acids. Okay, so you can see here on the diagram, we've got 2 strands of DNA, and on one of the strands, so maybe the 5' to 3' sequence, each of these triplet sequences goes on to make an amino acid. And then these amino acids would bind together, so we remove water, and they form that peptide link between them. And that's how we get our polypeptide. Okay, just a quick recap of the basic amino acid structure. We've got a central carbon, we've got a variable R group, we've got a carboxylic acid side chain, we've got a hydrogen group attached to that central carbon, and then we've got an amine side group as well. Okay, if you can't remember this, maybe go and take a look back over module 1. It should be in your brains now, it should be embedded in there really aggressively over the past 2 years, but if not, take some time, recap, that's fine, we can forget things, and we just got to make sure that they're back in our memories again. So now we're going to take a little bit of a look at mutations themselves, the different types and how they can affect protein synthesis. So definition wise, if I put a big arrow at the top, mutations are a change in the base sequences of DNA. Okay, and sometimes these changes of base sequences can disrupt the synthesis of proteins, so making those big 3D structures. And you can pause now and take time to consider how, or you can carry on playing and we'll go through it. So a mutation, we know, is a change in the DNA sequence for various reasons. We could have mutagens around, maybe it's just a part of mitosis, or a spontaneous change that's happened. And depending on that change in DNA, we have the potential, and it's not definite, that a different amino acid could be produced. Okay, now if a different amino acid is produced within a polypeptide sequence, that can start changing the primary structure or the primary sequence of our polypeptide. And we know from module 1 that it's the R groups of the amino acids, that make the tertiary structure. So if I've changed from maybe a small hydrogen, such as in glycine, and maybe turned into a big sulfur containing group in cysteine, that's going to have an effect on how my tertiary structure is able to fold up. Okay, so if we take this example of the glycine changing to cysteine, you can see that from that small glycine molecule, that might be a slightly compact part of the protein in its tertiary structure, but if it turns into a cysteine molecule with sulfur in there, maybe it can start undergoing things like disulfide bridge formation, and that's going to cause maybe a more open tertiary structure, which in turn can have an effect on the functionality of that protein. So maybe if it's an enzyme, for example, the active site won't necessarily be the same shape, and therefore it's not going to catalyse anything. So mutations can have various forms. We can either substitute bases, delete bases, or insert bases. And when I say bases, I mean one or more nucleotides. Okay, and if we're only inputting or deleting or substituting one nucleotide, we call it a point mutation. It is a single point of that genetic sequence that has changed. Okay, we don't call it a point mutation if it's more than one nucleotide. You need to be aware of this fact. Okay, it's not a negotiable point. It is a point mutation if one nucleotide is affected. Any more than one nucleotide, we don't call it a point mutation. It's just a substitution, deletion, or insertion. Okay. Now, the first type of mutation that we're going to take a look at is a substitution mutation, and for the rest of these mutations, what they are, as in substitution, deletion, insertion, is pretty self-explanatory. So for substitution, we are exchanging one or more nucleotides for another. So this is maybe swapping an A for a G for a C for a T. And there's 3 possible outcomes of a substitution mutation, and that is a premature stop. Okay, so maybe we say that the protein is truncated, and that just means that it's been shortened, or no more amino acids have been produced after that premature stop codon. It can affect the tertiary, and that should be above the 3 degrees, the tertiary structure, or it can be a silent mutation. And you can see in this diagram that what has actually happened is they've swapped out a codon. So they haven't just done a point mutation, they've swapped out the codon TGC for a GAT. Right, and we're going to take a look at each of those effects over the next few slides. So the first type of effect of that substitution mutation is that premature stop, and this is when the substitution mutation changes a base, so an A to a G, for example, to produce a stop codon in the middle of a sequence which would otherwise normally produce an amino acid. So as you can see, we've got this, the triplet table here, the degenerate sequence, and let's take an example of how this mutation or how a substitution can cause a premature stop codon. So if you imagine I've got the sequence T, G, T. Okay, now the way I use this table, we'll recap again quickly, is I look for the first base, so T's, and I'm looking at the top row, then I'm looking at G's in the second base sequence, So I'm looking down this side. And then finally, TGT is this top amino acid, and that's cysteine. Okay. Now, if I've got a point mutation that swaps this T for an A, so now my sequence is TGA, you can see that that is going to produce a stop codon. So if I've got a sequence that's like A, T, G, T, A, normally that would make A. So that would make lysine, and then it would be cysteine, and then another lysine. But if I've got that point mutation, then it becomes AAA, TGA, AAA, and that means that my sequence has lysine and then stop. Okay, and nothing is produced after that point. And that gives us that truncated or shortened protein. And because of this, depending on where that truncation or the shortening of the protein is, the protein that you make might not function. Okay, so if that stop is right at the beginning of our polypeptide sequence, you're not going to get much good out of that, are you? The second type of example that we're going to look at for the substitution mutation is where we're affecting our tertiary structure. Okay, now this is when the substitution produces a different amino acid from the codon. So you can see here that we've got the standard hemoglobin DNA sequence. There's just a section of it, so CTT, and that's going to go on to produce a glutamate amino acid. But if we've got this point mutation where the T has been swapped for an A, you can see that that is actually going to produce a valine amino acid, and that is going to affect the tertiary structure of my protein. You can see in this example that actually that single point mutation difference turns our hemoglobin from a normal functioning hemoglobin to sickle cell. And that means that the structure is slightly more shaped like a little scoop, and it's a structural and then a functional difference. It means that the hemoglobin can't carry oxygen as effectively. Okay, so this substitution mutation, especially if you've got a different amino acid being produced, is likely to have an effect on protein function if the protein itself is a functional molecule, so there's a biological use for it rather than a structural protein such as something like keratin. Now the final mutation, or the final effect of a substitution mutation, is a silent mutation, and this is where we've got that change of base, but we've still got the same amino acid produced. And this is because we've got a degenerate sequence. Okay, remember that means that we've got more than one triplet codon coding for the same amino acid. Now you can see here that for things like leucine, there are 6 different triplet sequences that code for that amino acid. Okay, and almost all of them, apart from methionine and tryptophan up here, have got more than one amino acid or more than one triplet sequence that codes for them. Okay, so oftentimes the point mutation won't actually have an effect. Okay, and we can say that the tertiary structure of the protein isn't affected by this mutation. There's no impact on functionality, and therefore we call it a silent mutation because we can't see any effect. Okay, and I want you to have a think about this question. Do you think the fact that DNA is degenerate is a survival mechanism for humans? So think about how these mutations arise. Think about why it might be good to have that degenerate sequence. Okay, you can pause, you can have some ideas, you can think about it, you can ask me some questions if you want, and then carry on when you are ready. Now, as I said, these are very commonsensical. Hopefully you've got some common sense. The second type of mutation is a deletion, and this is when a mutation causes a loss of one or more bases from the DNA sequence. Okay, and this gives us a change in the reading frame, or gives us a frame shift mutation. Okay, now if you don't know what a reading frame is, it's the ability of ribosomes to interpret a triplet, so a codon, of non-overlapping bases and turn it into an amino acid. That's a fancy way of saying that it's ribosomes reading threes. Okay, so if you imagine, we'll take this sequence, for example, I've got T, A, C, T, G, C, A, G, G, T. Okay, that is our sequence of DNA. We can put 5' to 3' just to be specific. And the ribosomes would normally read that sequence in the following groups. So TAA, CTG, CAG, GT, and there'd be something following on from that. Okay, whereas if we've got a deletion mutation, okay, and we'll again use that bottom sequence, for example, we've removed the T, and now we've got 5' TAA, CGC, GGT, 3'. And you can see now that the reading frame, or the way that we can read those threes, has differed. Okay, so actually every point after the removal of this T, so it's been removed or deleted, all of the subsequent nucleotides, so the G, the C, the A, the G, and the T, have all moved backwards. Okay, so they've just kind of buddied up and moved backwards to fill the gap where that T was. Now what that means is that the amino acids following on from that point of mutation could be different. Okay, so CGC might not code for the same amino acid as CTG did. Okay, and then that's going to have a maybe downstream effect on the structure of our protein. Ooh. So can you have a go, you can pause now, at reading these keywords that I've put here in purple, so base, codon, triplet, frame shift, reading frame, phenotype, alternative, tertiary, and functionality, and have a go at describing how deletion mutations affect protein structure. Okay, so you can pause. I'd say spend maybe 4 minutes on it, just to make sure that you're really using all of those keywords. There is no definitive answer for this, but I'll show you an example of how you might want to write it on the next slide. So pause now, have a go, and come back when you're ready. So here we've got an example of how we can use those keywords in sentences to give a nice overall scientific statement that helps us explain how deletion mutations affect the tertiary structure or the overall structure of a protein. Okay, so we're going to take this in turn, we're going to have a think about them, and we can discuss as we go. So a deletion of a base causes all other bases after the mutation to move in the reading frame. You remember in that previous example, we saw that we've got a sequence of nucleotides, perhaps A, T, C, C, A, and those are 3 codons read separately. And if we delete one of them, so I'll put a big red cross through maybe that C, then we end up with A still, but everything after that point of mutation has moved up one place. Okay, so that we say that the reading frame, the way that the ribosomes can then read those sequences, is changed, and that overall is known as a frame shift mutation. We have shifted the reading frame. Now the triplet codons after the mutation read by the ribosomes are going to be different, potentially. Okay, remember we could have silent mutations, but I mean if it's happening to all of the amino acids after that point of deletion, so the frame shift mutation, it's highly unlikely that the codons, all of them after that point of mutation, are going to be silent mutations. It's not going to happen. Okay, so the triplet codons read by the ribosomes after that point will differ, causing an alternative series of amino acids to be produced after the mutation point. Remember, if we look at this example, the AAA at the beginning is still the same. Okay, it's still present before and after the mutation. So everything we can say downstream of the mutation point. Okay, downstream just means at the later point. And that tertiary structure of the protein is likely to be significantly affected. It's not going to be a little bit different, especially if you've got more than one deletion. Okay, so maybe you've got two bases being deleted at two different points. And this is going to give us a change in the phenotype of the protein, the structural aspect of it, but also the functionality, the way that protein is able to work. Now we've got a little thinking example. I'd just like you to spend some time considering this, because sometimes the exam questions do like to try and trip you up. When would an insertion or deletion mutation not cause a change in the reading frame? You can have a look at the diagram at the bottom. Hopefully it should be maybe quite simple for you. I hope so anyway, because they flagged up the pictures or the changes nice and colourfully. Spend some time, pause now. You can spend about 30 seconds looking, thinking, maybe writing yourself an answer down, and then carry on when you are ready. So we don't get a change in the reading frame. We say it's a non-frame shift mutation if we insert or delete an entire codon, so a triplet sequence. And this is because we're just going to shift the reading frame one amino acid. Okay, it's not going to affect the amino acids that we produce, because we're effectively either inserting or removing one entire amino acid itself. But because we're changing the primary structure, we could still have an impact on the tertiary structure. It's just less likely to be as impactful as a point deletion. Now you can spend some time now finding out what each of these different types of mutation mean and how they affect the base sequences. So we can see here that we've got addition, duplication, inversion and translocation, and these are chromosomal mutations. I haven't got the answers for these. These are for you to do by yourself. So the pictures should help you, but you're going to need to do some research to find out what they mean and how they can affect the sequences of bases. Now we need to think about how these mutations actually happen. So we've got some biological causes of mutations that we know about, and then we've got another way that we can describe other things that induce mutations. Okay, so biologically, we know that mutations are a spontaneous event. Okay, they happen randomly during DNA replication and are permanent. So oftentimes, when DNA is undergoing replication, we're doing our checking maybe before mitosis. If the cell is found to be damaged or non-functional for any reason, maybe the mutation is really bad, then the body will normally just kill that cell, make it undergo apoptosis. But sometimes, especially if you think about things like genetic disorders, those mutations and those permanent changes to the DNA can persist in the body. Maybe they're a little bit hidden, maybe they're only on a small protein which the body goes, it should be fine. But actually, when that's developing into a fetus, a baby, actually it's going to have a massive effect. Now, outside of those spontaneous mutations that happen during mitosis, we've also got something called mutagenic agents or mutagens, which are things, chemical, physical or biological things that can cause mutations. Okay, and these are things that the scientists and stuff tell you not to do. So maybe you're into sunbeds. I hope not though, because they are bad for you. Lots of sunbathing, that UV is damaging to your cells. Maybe things like eating lots of red meat. That's been shown to increase your chances of mutations and therefore cancer. There's lots of different things that are going to cause mutations or increase the likelihood of harmful mutations occurring. Now, these mutagens are able to cause that genetic mutation that we've been discussing about. And generally, the government, the scientists are going to try to put rules, laws, legislation in to limit the impact of the harmful mutagens. So they're going to try and protect the population as best they can through laws. Even if you think about COVID now, we've got that legislation that everyone has to wear masks to stop the spread. Okay, so spend some time now specifically looking at how these two mutagens that are in green and orange are able to cause mutations. This is outside of the spec. It's important to know, especially if you're looking at maybe an A star and A in the paper 3 essay to have some really good, really detailed outside of the specification examples to show that you've done that extra research. So the two examples that I'd like you to look at, please, are high energy ionizing radiation, so alpha, beta and UV. Think about their sources, what do they do? And benzopyrene, which is found in cigarettes. So pause now, spend maybe 10 minutes or so looking at these two examples and finding out how they cause the mutations. Now, if you're in my class, if you've got access to the blue Oxford University book, have a go at that How Science Works Mutagenic Agents on page 239. Or if you're in the big double years, so year 1 and year 2 Oxford University book, it's on page 503. There's 5 questions, there's a little bit of reading to go with it as well. It might help you with the benzopyrene stuff. So if you're in my classes, take a look at the screenshot that I put on OneNote, and if not, then I'm sorry, I can't really help you at this point. Now we can think about mutations in terms of benefits and risks. So you might want to spend some time thinking now how mutations can be beneficial. And do the benefits outweigh the risks? So we spent a lot of time today thinking about how the mutations can cause things like a loss of function in proteins or a loss of tertiary structure. Is any potential good outcome of a mutation better than having all of that risk? And should, realistically, if there was some kind of higher power, should there be any mutations at all? So spend some time, you can pause, you can think about it, maybe make some notes, do some research, and then carry on when you're ready. So, for the most part, benefits of mutations are there to increase the genetic diversity of a population. Okay, so if you think about it in terms of Darwin's finches that we will have looked at in Module 7, the mutations that happened in each of those finches' beaks are going to make them slightly better suited to their environment. Means that they're going to be able to adapt, to change, to overcome. And in some cases, mutations can be really beneficial in terms of giving people immunity. So for example, there's a point mutation called CCR5 delta 32, which prevents the formation of full-sized receptors on the outside of the cell surface membrane. And that means that HIV molecules are unable to bind and infiltrate the host cell. And that effectively gives rise to immunity from HIV. It's very, very unlikely that you've got that point mutation, but it's not to say that you haven't. I wouldn't recommend testing it though, it's probably a bad time to risk your health. Conversely, the risks mean that every mutation is almost always going to be harmful for some aspect of the individual. And that means that the organisms are going to be less well suited to the environment. So if you think about maybe things like people with albinism, for example, so they lack the pigment in their skin, that mutation that means that that pigment is not present isn't very helpful, because normally in our skin, the pigments offer a protection against the UV rays from the sun. So the pigment, or melanin, overlays the nucleus and acts as like a little shield to stop the rays being able to hit the DNA and cause that mutation that is going to maybe give rise to skin cancer. But if you've got albinism or you're an albino, you're not going to have that melanin, that protective shield. Okay? And that means that your DNA is slightly more at risk of being hit by the UV rays. And that's going to mean that you're realistically less well suited to the environment. It means you're more at risk of certain conditions. And there's lots of examples of this in nature. Albinism is just the first one I could think of. Okay, you could do some research maybe to see what other examples, what other exciting human examples have got where people have mutated to be less well suited to the environment. Now, if you're in my class and we're doing this as part of an online learning lesson, some of you it'll be revision, have a go at the summary questions on pages 238, that's in the Oxford book, and then have a go at either planning or writing an essay on one of those topics. So these two titles, A and B, are the examples of essays that you might get in paper 3. And you can see that they're very, 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 very unspecific. They're quite broad. Control of processes in cells and the importance of these controls, and the causes and importance of variation and diversity in organisms. So you might want to talk to your teacher, me, or whoever you've got around you on how's the best way to start these, maybe look into planning it. There's lots of resources on the AQA website as well. If you search AQA 7402 essay prep, then there should be lots of files to help you and show you what kind of mark scheme and stuff, or what kind of guidance the markers get for these essays. So make a note. You can have a go at those if you're in my class, if I put the screenshot on there. Otherwise, we're going to take a look at some exam questions. In a recent medical trial, 62 children with this genetic mutation, I'm going to assume something about asthma, were studied. Their asthma was not controlled well by someterol. 31 children continued using someterol, and the remaining 31 were given an alternative drug, montelukast. Montelukast is not routinely prescribed because someterol is far more effective for most children with asthma. After 1 year, the children taking montelukast had better control of their asthma and were able to reduce their use of montelukast. Suggest why these children responded better to montelukast than someterol. So you can pause, it's 2 minutes, maybe give an example, suggest, so give reasons for using your knowledge. But you're not expected to know the answer. So you might want to think about what you know about mutations. Why might someone respond better to a drug of one type than another? 3 minutes, pause, and carry on when you're ready. Were that, after 1 year, the children taking montelukast had better control of their asthma and were able to reduce the use of montelukast. Comment on the reliability of the results of the medical trial. So the result of montelukast. Okay, so reducing the use of that drug. Comment on the reliability of that result. And remember, reliability is to do with repeatability. It's to do with, are you going to get the same result over and over again? So that's 1 mark. It should take you about a minute and a half, hopefully. And for part 3, it is proposed that a simple saliva test could identify those children who have the mutation. What would be the source of genetic material used in this test? So it's a saliva test. What would be the source of genetic material in saliva that they could use to test? So again, that's 1 mark, maybe a minute, 2 minutes. You might want to spend a little bit more time thinking about that one. Here's a completely separate question. Meiosis is a source of genetic variation. Mutation is another source of genetic variation. What feature of the DNA molecule is changed as a result of mutation? And then part 2, discuss the possible effects that mutation can have on the structure and function of a protein. So the first question about what feature of the DNA molecule is 1 mark. Spend some time, maybe a minute or so. Should be quite simple. You're just looking at a part of a structure of DNA. And then for the second point, discuss the possible effects. That's 3 marks. You should take about 5 minutes for this. Okay, asked about the possible effects. So you can list maybe the 3 things that could happen depending on how that mutation is. So pause now, have a go, and then when you're ready, the next few slides are the answers. So the first question that we looked at was asking why those with a mutation might be able to respond better to that montelukast versus the someterol. And you could have said something as follows. There's 2 marks maximum. Each new line is a new mark, or whenever you see a semicolon after that is a new mark. So there's 4 marking points here. The mutation resulted in the receptor, so the part of the cell that's going to bind to the drug, having a complementary shape to montelukast. It's no longer complementary to the salmeterol. The montelukast is therefore able to bind where the salmeterol can't. Okay? Or you could have also said that the montelukast has a different receptor. So you're giving reasons as to why one drug might work better than the other. For the second question, we're talking about reliability, and this is a good time to be doing maybe an individual, a human studies subject, so maybe psychology or sociology, because it's asking about the reliability of a sample. So you could have said that it's either not reliable because the sample size is too small. Okay, notice that 2 is in bold because it's implying that it's too small to be able to get a result from. Or you could have said that there's only 62 children in the study. The idea is that it has to be comparative. So just saying that the sample size is small, well, yes, it is, but it's too small for the results to be reliable. It's not that there's 62 children, it's that there is only 62 children there. Again, it's implying that it's too small to be useful. You could have also said that it is reliable though, because 31 is quite a large sample, or that 31 is a suitable number for a phase 1 trial. So we're initially studying the effect of that drug in a group of ill individuals, and we only need a small sample size for that. We're trying to see if it's going to kill anyone, basically. So if it's a small sample size, you know, there's less people to kill. And the final question, talking about the saliva sample and what kind of source of genetic material there would be, you could have said cheek cells, cells lining the cheek. You might have also said white blood cells in the saliva, or the salivary gland cells. And notice that saliva is not a source of DNA, because saliva itself is not cellular. It's made of like liquid and salts and things like that, not cells. The cells are going to come from surrounding tissue, so your cheeks, white blood cells, or salivary glands. The penultimate question was looking at the structure, or which part of DNA is changing, and this is about the sequence or order of bases. You could have said the order of nucleotides as well, you could have said nitrogenous bases, but it was about the DNA structure. So if you've mentioned something about the amino acid sequence, that is not correct, not right. The final question was looking at the possible effects of mutation on the structure and function of a protein. So the first marks are pretty standard, that you get a different primary, secondary, or tertiary structure. You could have also said for this one that you get a different sequence or order of amino acids as well. And then it asked you about the effects. So you could have said that the protein becomes shorter due to deletion or a stop codon. So you could have said it's truncated. You could have said that it's also longer due to insertion. So maybe we've added in some bases, maybe we've added in a few nucleotides. You could have also said that the protein remains unchanged due to a silent mutation, or maybe you said that a mutation can be silent if the nucleotide that's mutated just produces that same amino acid, so we can't really see an effect. And you could have said that the function is going to be lost or worse or better, so we get a loss of function, the protein doesn't work as well. If it's functional, it doesn't have the same structural properties if it's a structural protein, and overall we're going to get a significantly different, normally, effect. So I hope you found this useful. If you've got any questions, let me know, and yeah, I'll see you next time. Bye.